Hello boys, welcome back to another interesting session of video tutorials, and this session is meant for class 12th biology students, and this is for 8th September 2020. We continue from where we have left, and this, as I said, has been the baby of the biology, and this baby has lot of potential in future. And basically we were having an aim, a larger aim of creating a healthy society, a healthy nation, because a healthy society and healthy nation can be created only when our biology plays a crucial role. What is that? The biology has to make our people living in the society healthier. Once people are healthier, society is healthier and the nation is healthier. A healthy nation always does wonders. Look at the corona now, COVID-19, it is a reverse process. And this is creating a lot of sick nations. And you must have seen in European countries, heavy losses of valuable human lives. So in order to stop those, the biology has to take the front step. And thanks to this baby called biotechnology. Biotechnology is the only answer for such COVID-19 situations. So here we go for that. Recombinant DNA, that is the only solution. We need to make rDNA, and once this rDNA technology is made, we will be very healthy nation. So basically we were talking of the various steps involved. I think yesterday we have completed this isolation of genetic material in the DNA. We used varieties of enzymes. I just wanted you to remember lysozyme, cellulase, chitinase, protease, ribonuclease. Remember, all these enzymes are used in isolation of genetic material DNA. I also promised you that once you come back to school, you will be performing the experiment. And then second step, fragmentation of DNA by restriction endonuclease. The moment you hear this restriction endonuclease, you must remember the plasmid DNAs, particularly artificial plasmid DNA. What is that? HBr 322, right? And PBr 322, right? Plasmid. And then we also know that DNA ligase enzyme. And remember the sticky ends of two DNAs, and we call the process annealing. And this annealing will produce rDNA. Then isolation of desired DNA fragments, gene of interest, we started isolating. And not only isolation, thanks to Carey Mullis in 1985, he has used the method PCR, polymerase chain reaction, with which we can amplify the gene of interest. And in that we were discussing 3 steps: denaturation, annealing, and polymerization or extension. Now in this, we are going to talk about this step number 5th, transferring. Of course, ligation we have done already, and that is joining of the pieces, and transferring of rDNA into host. How does that happen? And so basically the insertion of rDNA into host cell or organism. So basically the vector DNA or plasmid DNA and then foreign DNA carrying gene of interest are cut by the same restriction endonuclease to produce complementary sticky ends. With the help of DNA ligase enzyme, the complementary sticky ends of two DNAs are joined, we said annealing, to produce rDNA. Eukaryotic genes do not function properly when cloned into bacterial cell because of introns. In such cases, DNA is made from mRNA by reverse transcription or synthesized artificially. This rDNA is inserted into host bacterium by, we said, transformation using cold CaCl2, calcium chloride solution. Of course, you will also be using in our lab the cold CaCl2. Okay. And the bacterial cell containing the desired rDNA is selected using selective antibiotic in the culture medium. So this is what we were talking of yesterday. And further, I said you will be doing this experiment in the class once you come back to schools. And we were also talking of the cutting of the DNA at specific locations and then joining the gene of interest, DNA ligase we said. Remember here, this is how we are doing it, ligation of gene of interest, and then amplification of gene of interest. This also we discussed, 3 steps we discussed, and gene of interest is polymerized in this fashion, and there are conditions, particularly remember temperatures. And then now we come to insertion of recombinant DNA into host cell, and the host cell should be competent. Look at that, we should make the host cell competent. So insertion of our DNA is possible only by making the host competent. This is achieved by chemical treatment, micro injection, biolistic gun or gene gun, and disarming pathogen. These are all the things we must do so that the host becomes competent. The phenotype of organism alters when rDNA is transferred to host. Example, rDNA with gene resistance against antibiotic ampicillin. Remember, ampicillin R we wrote in that PBR322, ampicillin resistant gene. So it is transferred to the host E. coli normally, and phenotype gets altered and resistant against ampicillin, that becomes a transformant. When plated on agar plate with ampicillin, transformants will grow with the change in phenotype and non-transformants will die. There is a small experiment also in this. So presence of ampicillin resistance gene selected transform cells and considered as selectable markers. So we have got what is called selectable markers. And what is that experiment we do? This is how we do. So we have got a transformed cell, we have with the plasmid with gene of interest, and non-transformed cell. We put this in nutrient medium plus ampicillin. Overnight if you see, ampicillin resistant colonies would have developed. So this is what the small experiment that can be performed. And once we get this, now our target, what is the final target? Obtaining the foreign gene product. What is the product? It is we are expecting is recombinant protein, because rDNA, recombinant DNA. So recombinant DNA will give us recombinant protein, and this is what our final target. And proteins with various functions we obtain through this process. So alien DNA binds with cloning vector rDNA transferred to suitable host. Okay, this is the step number 1. rDNA expresses itself, that is target protein, under appropriate optimal conditions, and after cloning of rDNA to particular number of copies. And target protein, what is that we need? In lakhs, in crores, high quality we require. And heterologous host, if protein encoding gene is expressed, then we get recombinant protein. And heterologous host, gene or gene fragment that does not naturally present in host, the gene expresses itself, recombinant protein we call it. Cells with Haber clone genes of interest grown in laboratory in small scale. Of course, in laboratory we will be growing in small scale, but what about for society? We need in large scale. So culture of dividing host used to extract desired protein, then purified by different separation techniques. It is not simply just obtaining the desired protein. We need to procedure or follow standard procedures so that the protein that is formed will be purified. So in large scale, cells can be cultured in a continuous culture system. Here the used medium is drained out from one side and fresh medium added from other to maintain the cells. So physiologically, most active exponential phase will take place because we wanted in larger quantity. This culture method to produce large biomass leading to higher yields of desired protein, desired gene or gene of interest, and desired protein. And what is biomass? Already you know, biological material derived from living or recently living organisms. To produce large quantities of products, we need special structures. They are called bioreactors, and these are having a volume of 100 to 1000 liters. and large volumes of culture is processed. So bioreactors are the vessels in which recombinant organisms like microbial plant, animal cells, human cells, and culture medium with ambient conditions, so biologically converted into specific products. What are the specific products? It is the recombinant proteins or enzymes. So optimal growth is achieved with growth conditions. So there are conditions apply. What are those? Optimum temperature should be there, optimum pH levels, volume of the substrate, various salt concentration, vitamins, and most important, oxygen supply should be there for these bioreactors. So bioreactors are considered as vessels in which raw materials are biologically converted into specific products by microbes, plants, and animal cells and their enzyme. So these are the bioreactors. And what is the structure? There are two broadly used bioreactors. They are stirring type. They are simple stirred tank bioreactors and sparged stirred tank bioreactors. So commonly used is stirring type, and cylindrical. They are basically with the curved base to facilitate mixing of contents, equipped with a stirrer, the one that mixes, even mixing, and oxygen availability throughout the reactor. Oxygen delivery system is there, bubble air through reactor, and there is an agitator, thorough mixing of the contents. Agitator will ensure that. Also have foam control system, temperature control system, pH control system, and sampling port. What is sampling port? It helps us to withdraw small volumes of culture periodically to check the progression. So these are the various control systems that will be present in the bioreactors. Here we come. These are the bioreactors will be there. You can see here it is a flat base, and here you can see the curved base. And so we have got what is called simple stirred tank bioreactor. This is simple stirred tank bioreactor, and this is stirred tank bioreactor. A stirred tank bioreactor is usually cylindrical, as you are seeing, with a curved base to facilitate the mixing of the reactor content. The stirrer facilitates even mixing the oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. The bioreactor has an agitator system. You can see the agitator system. It has got oxygen delivery system, oxygen delivery system, foam control system, temperature control system, pH control system. All these systems are inbuilt. And sampling ports, we have got that sampling ports, so that small volumes of the culture can be withdrawn periodically. After completion of the biosynthetic stage, the product has to be subjected through series of process before it is ready for making it as a finished product. The process include separation, purification, and these separation and purification methods together are collectively referred as downstream processing. So the product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials. As in case of drugs, strict quality control testing for each product is also required. Then only we get that the final product marketing can be done. So basically, downstream processing. As I said just now, downstream processing refers to the recovery and purification of biosynthetic products, particularly pharmaceuticals, from natural sources such as animal and plant tissue or fermentation broth. Products obtained from the bioreactors subjected to series of process before marketing as finished product, as I said just now. This process includes two steps: separation and purification of biological products. Products are formulated with suitable preservatives, then should undergo thorough clinical trials like drugs, like how normal tablets, injections are being treated, like that. Strict quality, this is important because there can be an unseen dangers, unexpected dangers can be there. So to avoid that, strict quality control testing is required for the final biological product. So this is how we can summarize the whole thing. What is that? So we have got now step number 1, isolation of plasmid DNA and DNA containing gene of interest. Look at this, cell containing gene of interest. This is DNA chromosome, so that's obtained. This is gene of interest, and we are at the same time selecting E. coli. E. coli will have bacterial chromosome and plasmid. We are interested in this extra chromosomal circular DNA that will be extracted out by using various enzymes, and this is cut through the restriction endonucleases, and the gene of interest also is cut using restriction endonucleases, and using ligases. Look at this, gene of interest can be joined to this plasmid, so gene inserted into plasmid, and once we obtain the rDNA, plasmid put into bacterial cells, suitable host we set, and once you can see here gene of interest is incorporated, this becomes a recombinant DNA, and the bacterium is also called a recombinant. Here see we are using only E. coli, here we are using recombinant E. coli. Now cells cloned with the gene of interest, now these numbers are to be increased, and identification of a desired clone, and once that is done, we can have varieties of things can be cured. Like for example, once the gene of interest is increased, the gene can be used in plants, gene for pest resistance inserted into the plants, so that we need not spray any chemical there. Without using any chemical, with just insertion of gene of interest, we can make the plants disease free. Similarly, we can also use this for research, basic research of a gene, and also we can use the gene of interest to alter bacteria for cleaning up toxic waste. We know this is the biggest problem that every country is facing in the world. What to do with the wastages, whether it is e-waste or bio-waste. So the gene of interest can be used to clear off this waste, toxic waste, and you know the industrial waste, how much it is causing havoc in cities, and the drainage, sewage. So all this can be cured. Just imagine as this baby grows and becomes an adult, how beautiful this world will be. And similarly, copies of protein, once we obtain the R protein, recombinant protein, that can be used for making a dwarf fellow, stunted the children to grow tall. So naturally now many people with the stunted growth will be doing lot many exercise, and sometimes they get duped by some advertisements by fake doctors that they would make them tall within days or months. So that can be done. And then basic research, we don't know much, still we need to understand what is the behavior of the protein. So for basic research we can use it. And then protein dissolves blood clots in heart attack, so without surgery we can clear off any clots, any blockages in the heart. So various applications are there, and we can make this whole nation, whole world healthy with disease free, just by making disease free. And that is the aim of biotechnology. What a good aim, is it? So we need to encourage